So my name is Tanya Khokhlova. I'm from the University of Washington, and I'll be giving an overview of immunological effects of boiling histotripsy tumor ablation in available preclinical studies. So boiling histotripsy is a regime of pulsed HIFU that results in mechanical soft tissue fractionation with not discernible thermal effects. It looks grossly like this, and histologically it looks like completely fractionated tissue down to subcellular level. It is mediated by HIFU pulses that are on the order of a few milliseconds long and contain shock fronts of sufficient amplitude to produce vapor bubbles at the focus. Within a few milliseconds, the interaction of those vapor bubbles with the remainder of the shocks causes cavitation, atomization, and tissue fractionation into subcellular debris. So hypothetically, what can this regime do to alter immunological status of a tumor following its ablation? So I borrowed this very helpful diagram from this review, and each of those aspects has been to date demonstrated in one or the other preclinical study. So following boiling histotripsy, all the intracellular or extracellular antigens contained in the tumor are released into extracellular space, which increases the access to tumor antigens and promotes a cell infiltration into those antigens. In addition, it causes the elevation of damage-associated molecular patterns, or DAMPs, that cause infiltration of lymphocytes into the tumor and tumor-presenting cells that triggers the inflammation cascade, cross-presentation in the lymphatics, and eventually T-cell recruitment and proliferation at the tumor site, and transition of sort of cold or immunosuppressive tumor microenvironment into hot microenvironment. And to date, the effects of boiling histotripsy ablation were explored in a few preclinical models, and I listed them here in the order from relatively more immunogenic or relatively more hot, like melanoma and renal cell carcinoma, to very cold and less immunogenic, such as pancreas cancer, colon adenocarcinoma, and neuroblastoma. Boiling histotripsy has been deployed with and without combination with immune checkpoint inhibition, and I'll cover the general findings and dynamics of the immunological processes that take place in each of those tumors. So starting with melanoma, this study has been conducted at by our group almost a decade ago, in which B16 melanoma was subcutaneously grafted and nearly completely ablated with boiling histotripsy. You can see the effect of the treatment here. Following the ablation, tumor outgrowth and immune cell populations were characterized in the tumor, blood, and lymphatics. And while this ablation did confer the retardation in tumor growth, the residual tumors still grew. And in terms of immune cell populations, there was disappointingly no statistically significant difference in the number, phenotype, or activation of any of the T cells or maturation of dendritic cells. However, there was a twofold difference in the number of MHC2-bearing cells in the spleen and lymph nodes, which indicated that there is an inflammatory process going on. However, it wasn't enough. And in light of more recent studies, that result now makes a lot of sense because given the recent successes in checkpoint inhibition in melanoma, the inflammatory cascade isn't enough. T cells that are infiltrating need to also be not incapacitated by the tumor. In an even earlier study from Duke University, they looked at treating colon adenocarcinoma subcutaneously grafted and treated with either boiling histotripsy. It was not even known as boiling histotripsy at the time. It was called mechanical HIFU or thermal HIFU, and compared the effects of the two. And they characterized in more detail the short-term effects of those treatments, and found that it was that the inflammatory effects were quite rapid, in particular, in the case of mechanical treatment, the infiltration of dendritic cells happened in 1 day, and 2 days later, they were gone, followed by a substantial increase in tumor-draining lymph nodes. And further on, about a week later, increased interferon gamma production by tumor-specific T lymphocytes in the spleen. And those treatments did retard tumor growth and challenge tumor growth. However, the tumors still grew. So the effect was not complete, but that is a familiar theme now. In a more recent study done in renal cell carcinoma, also from our group, we used a bilateral kidney tumor model, and it's a spontaneously arising kidney tumors in genetically modified ECHO-rat model. And one of the tumors was partially ablated with boiling histotripsy, and we've shown that those processes that I illustrated earlier, the release of DAMPs, notably HMGB1, does occur within short term, and circulating and intrarenal pro-inflammatory and anti-inflammatory cytokines do get elevated transiently. And more importantly, intratumoral infiltration of CD8 T cells was observed bilaterally, so not only in the treated tumor, but also in the contralateral one. Then when we looked at it more long-term, and we wanted to see if all of those effects are tumor-specific, or would they occur simply due to trauma to the kidney if we ablated just part of the kidney. And so we treated either the tumor in the ECHO-rat or just the kidney in a wild-type rat, and survived them for longer, 7, 14, or 56 days. And notably, those tumors are fairly slow-growing and they don't metastasize, so we had the luxury of doing that. And so the lesion evolved over 8 weeks, and by 8 weeks, it was almost completely resolved with minimal fibrosis. And similar findings were seen in the kidney. And in terms of the population of immune cells, importantly, we found that there were more CD4 effector memory T cells in tumor-draining lymph nodes and CD8 effector memory cells in spleen and lymph nodes, only in the ECHO-rats and not in the wild-type rats, which indicates that indeed this effect, not only is there evidence that there's longer-term adaptive immunity generated, but also it's tumor-specific. The intrarenal cytokines mirrored the cellular changes that I mentioned earlier. Again, they were mostly observed in ECHO-rats and not wild-type rats. And importantly, there was over a 10-fold increase in IL-2 concentration at 8 weeks in ECHO-rats, but not wild-type rats. And that's important because IL-2 is an immunotherapeutic that has been used to treat renal cell carcinoma in the past to some success. And a very important recent study in boiling histotripsy was done at by a group at Children's National in a neuroblastoma tumor. They treated large unilateral neuro2 tumors in mice, and they only ablated a very, very small fraction of them, about 2%, and combined it with immune checkpoint inhibition therapies, notably following the HIFU ablation in only 3 doses, 1, 3, and 6 days following treatment. And this graph shows the percent survival of the mice with all the different permutations of treatment groups. And it can be readily appreciated that only the group that combined all the treatments together had substantially improved survival, and HIFU alone did not produce as much of a survival benefit, which only makes sense if we look at PD-L1 expression by the tumor 72 hours after HIFU. So the untreated tumor doesn't express PD-L1, whereas the treated one does, and about 3 days following HIFU. So providing immune checkpoint inhibition annihilates this immunosuppressive signal and allows for this sustained survival. So they also looked at immune cell populations and circulating cytokines short-term, and the findings were similar to the previous works in that there was an increase in tumor-infiltrating T cells and dendritic cells, increase in dendritic cells and tumor-draining lymph nodes, and short-term or sustained increase in interferon gamma, and while decreasing IL-10, which is an immunosuppressive cytokine. Even more importantly, they found abscopal effect in bilateral tumor model following this therapy, this combination therapy, and they found it to be mediated by effector memory T cells. And moreover, they found to be adoptively transferable and conferring protective effect against rechallenge. Last but not least, there's a very recent study from a group at the UK done in orthotopic pancreatic cancer model, KPC tumor. And I have it with an asterisk because they talk about it as boiling histotripsy-like treatment, but by all signs of it, it is mechanical fractionation, so I included it here. It was also combined with CTLA-4 and PD-1 inhibitors. And in contrast with the previous study, the immune checkpoint inhibition was administered every 3 days throughout the observation period. And that combination also makes sense because, as we can see here, KPC cells do express PD-L1. And again, we can appreciate that the improved survival was only associated with when HIFU was combined with immune checkpoint inhibition. The improvement in survival was definitely more modest than what we saw just before in neuroblastoma model. However, one has to appreciate that pancreas tumors are notoriously difficult and arguably some of the coldest tumors there are. And so even with very little ablation of the tumor, very large established tumors, this is actually quite an impressive effect. And what they saw was that interferon gamma producing CD8 T cells were increasingly infiltrating into the tumor 2 and 12 days post ablation. And the immunosuppression was not increased, at least by boiling histotripsy. And interesting and intriguing effect they found is that decreased frequency of CD8 T cells and tumor draining lymph nodes following ablation, which they hypothesized could be due to disrupted lymphatics. And that's an effect that we should be looking at potentially in the clinical translation, as that's something we'd probably want to avoid. So in conclusion, boiling histotripsy mechanical ablation of tumors does promote lymphocyte infiltration and pro-inflammatory tumor milieu, even if partial, and that process is quite rapid on the order of days. So if that milieu is sustained, for example, with checkpoint inhibition or other therapies, tumor-specific effector memory T cells may be formed, and adaptive immunity may confer tumor regression and abscopal effect. However, in particularly cold tumors like pancreas cancer, this may not be enough, and additional immunotherapies, in particular, perhaps there's a role for CAR T therapy or other adoptive transfer approaches could make sense. Thank you.